Very good morning to one and all. Today's topic for the lecture is enzymes used in genetic engineering. Prior to know about the enzymes used in genetic engineering, it is mandatory to know something about genetic engineering. Genetic engineering, as the word suggests, it is made up of two subwords, that is genetic and engineering. Genetics means related to genes and engineering means the manipulation. So what is genetic engineering? Genetic engineering is the manipulation of the genes in order to obtain the product of our choice of interest. What are the basic steps of genetic engineering? In genetic engineering, first of all, we isolate the DNA fragment that is to be uploaded and that is to be checked for the expression of our desired product. Then we upload it into the vector molecule. What are vectors? Vectors are the molecular vehicles. Then finally we upload it into the host and finally we check the expression of the desired protein. What we require a list of number of enzymes in order to accomplish our aim in genetic engineering. The term genetic engineering was coined by Paul Berg in 1972, who is known as the father of the genetic engineering. Then moving on towards the main topic, that is the enzymes used in genetic engineering. These are some enzymes which are used in genetic engineering: restriction endonucleases, alkaline phosphatases, polynucleotide kinase, lambda exonucleases, E. coli exonuclease third, S1 nucleases, clinofragment, RTAs, RNAs, terminal deoxynucleotidal transferase, and a lot more. We will start with restriction endonucleases. As the name suggests, restriction endonucleases. Endonucleases is the class of enzymes which cleave the DNA randomly at any site, and restriction endonucleases are the class of endonucleases which cleave the DNA fragment or RNA fragment at a particular site. That is, these are the endonucleases which are restricted to act upon a particular site. Those sites which are being recognized by restriction endonucleases are called as recognition sites. There are mainly 5 types of restriction endonucleases, namely type 1 restriction endonuclease, type 2, type 3, type 3S and type 4 restriction endonucleases. Depending upon the type of or the number of base pairs which are being required to recognize the restriction endonucleases, their particular recognition site. There are different factors which types of restriction endonucleases require to maintain their functioning. That, for example, few of them require magnesium as the cofactor, few of them does not require magnesium as the cofactor. The mandatory requirement which every type of restriction endonuclease require is the need of the ATP, that is adenosine triphosphate. Then we move towards the nomenclature of restriction endonucleases. How these are named? We know example, we will study with the help of the example. Hind third. This is the name of a typical type of restriction endonucleases. How it is named? The first letter of the name of the restriction endonucleases describes the name of the genus of the bacterium from which these restriction endonucleases have been isolated. Here H recognize the Haemophilus. Next, 2 subletters, that is IN, represents the initials of the species of the bacterium from which the respective endonuclease has been isolated. IN is from influenzae. That is, this restriction enzyme has been isolated from Haemophilus influenzae. And D and the last Roman word represent the strain and the number of the restriction enzyme that is being isolated from Haemophilus influenzae. Then what about the recognition sequences that are being recognized by restriction endonucleases? Restriction endonucleases recognize a particular class of the restriction sequences which we called as palindromes. Now palindromes could also be of 2 types: simple palindromes and palindromes with rotational symmetry. What are simple palindromes? Simple palindromes are those sequences which are being recognized by restriction endonucleases which have the mirror image of adjacent nucleotides within the same strand. For example, we have this particular nucleotide sequence, then its palindrome will have the mirror image within the same strand. If we put mirror in between, so these are two respective mirror images of each other. Such type of sequences are known as palindromes. Now what are palindromes with rotational symmetry? Palindromes with rotational symmetry are those recognition sequences which are being identified by restriction endonucleases in which the mirror image is in another leading or lagging strand of DNA. For example, if we have this sequence, then its mirror image will be present in its counterpart strand. And so on. What are the cleavage patterns that are being produced by restriction endonucleases? We all know that restriction endonucleases are the class of endonucleases which cleave the DNA or any biomolecule at a particular site. After the cleaving, they produces two types of patterns, that is even ends or staggered ends. Even ends or sticky ends rather. What are even ends? Even ends, suppose we have a certain sequence which is being cleaved by the restriction endonuclease. What will be the type of cleavage pattern that is known as even end will be, suppose this site is being cleaved by restriction endonucleases. So the number of nucleotides present on left and right side are equal in even ends. Example, this. We have 3 nucleotides on the left and 3 nucleotides on the right. Such type of cleavage pattern is known as even ends produced by cleavage of restriction endonucleases, and such ends are known as even ends or the blunt ends. What are sticky ends? Sticky ends or cohesive ends are the irregular ends or the irregular pattern that is produced after the action of the restriction endonucleases. For example, this is the strand which is being cleaved by restriction endonucleases having restriction site over this. So what will be the pattern produced? This. So we have 1 nucleotide on this side and around 5 nucleotides on this side. This produces an irregular pattern that is called sticky ends or cohesive ends. This was all about the restriction endonucleases, their types, their nomenclature, their recognition sequences, their cleavage pattern. Then moving on to next type of enzyme that is most frequently used in genetic engineering is alkaline phosphatase. As the name suggests, phosphatase. Phosphatase is made up of 2 subwords, that is phosphate plus ase. Ase means to hydrolyze something. So alkaline phosphatase is the enzyme that is being used in genetic engineering in which we remove the phosphate group from 5' or any end of the DNA. Then what is polynucleotide kinase? The main function of the kinases is to add up a certain group to the biomolecule. What it does, it adds up the phosphate group to the 5' end. Lambda exonuclease enzyme, it removes the phosphate group from 5' end. E. coli exonuclease, third, as the name suggests, it is the type of exonuclease which is being isolated from E. coli, having the third type of invention. What it does, it simply removes the nucleotides from 3' end. What is the S1 nuclease? S1 nuclease is also a part of enzyme, as the restriction endonucleases, which normally are known as molecular scissors, cleave at a particular site. S1 nuclease also cleaves the DNA, but only at the single strand. This is the main difference between S1 nuclease and this restriction endonuclease. Then what is the clinofragment? Clinofragment is the part of DNA polymerase 1 enzyme which helps in production of the protruding ends or in the even ends. RTase as the name suggests, reverse transcriptase. This was the enzyme which was discovered by Tamin and Baltimore. We all know about transcription. What is transcription? Conversion of DNA molecule to RNA molecule. Okay. And what is reverse transcription? Converting back of RNA molecule to DNA molecule. This step is accomplished with the help of enzyme known as RTase, that is reverse transcriptase, the enzyme which is useful in the function of reverse transcription. What is RNase? RNase is the enzyme which is used in hydrolysis of the RNA molecule. Terminal deoxynucleotidal transferase, it adds up the nucleotides to either of the ends of the polymer. That's all about the enzymes used in genetic engineering. These are the few that are enlisted here. There are a number of enzymes such as DNA ligase, DNA polymerase, RNA polymerase, which function in natural way as well as in the artificial way to accomplish the fate of the genetic engineering. So, for example, DNA ligase, which is commonly known as molecular glue. What it does, it simply attaches the nicked fragments of the DNA or RNA. That's all for the today. Thank you.